wheat analogy for protein synthesis. Transcription. That is, from blueprint to seed. Imagine a farmer has a detailed blueprint for a specific type of wheat, in this case which is DNA. This blueprint contains all the genetic information, such as the color, size, and nutritional value of the wheat grains. Plowing the land. To start, the farmer plows the land, that is, unwinds the DNA double helix to access the specific gene, a section of the blueprint, needed for the desired wheat. Sowing the seed. Using the blueprint as a guide, the farmer sows seeds, that is, transcribes the genetic information from DNA into mRNA. This mRNA is a single-stranded copy of the gene that can leave the nucleus and travel to the cytoplasm, like a portable blueprint that can leave the office where the master plan is stored and travel to the field where the wheat will grow. Translation. From seed to grain. Harvesting the crop, the mRNA, in this case, the seed, travels to the ribosomes like small factories in the field. Here, the processing begins. Processing into flour. At the ribosomes, the mRNA blueprint is used to assemble the wheat grains, which are proteins from smaller building blocks called amino acids. This is like processing the harvested crop into flour. Refining the flour. The process of assembling the protein from amino acids is like refining the flour. The order in which the amino acids are linked together is crucial, just as the type of flour used determines the final product: bread, cake, etc. This order is determined by the information encoded in the mRNA blueprint. Protein modification. The bakers, once the flour, in this case the polypeptide chain, is produced, it's not always ready for use. It often needs further modifications, just like flour needs to be mixed with other ingredients and shaped into a final product. Mixing and blending. This stage involves adding chemical groups, like sugars or phosphates, to the protein, folding it into a specific three-dimensional shape, and even cutting it into smaller pieces. These modifications are crucial for the protein to function correctly. Shaping and decorating. Different proteins have different functions within the cell. Some act as enzymes, catalyzing chemical reactions, some transport molecules, and some provide structural support. These modifications help shape the protein and give it the specific features needed for its particular job. In summary, just like wheat production involves a series of carefully orchestrated steps, from sowing the seeds to harvesting and processing the grain, protein synthesis is a complex multi-step process that involves transcribing genetic information, translating it into proteins, and modifying those proteins to fulfill their specific roles within the cell. If you like this video, please subscribe and share.
Generated